Hi, my name is Cedar. I'm a professor emeritus at the Hebrew University in Jerusalem in the medical school. And I'd like to tell you a little bit about our research over the unbelievable time of about 40 or 45 years. And so our research is interested in or concentrates on something called epigenetics. And the principle is very simple. All of us are built from parts, like a big Lego item. And we have many parts in our body. And the information from making those parts comes from our genes. The genes are written into DNA in the chromosomes. And this information gives the cells it serves like a book or an encyclopedia for the cells and tells them how they should build the different proteins, the different pieces of Lego to put together the entire body. What we discovered about 40 years ago is that in addition to that basic textual language, there's another language attached to our genetic material. It's an annotation. It's an annotation, just like you would annotate a text in English or in Hebrew. The cells are able to annotate the book of DNA, the book of genetic information. And this annotation, of course, is not with a pencil or a pen, but it's done chemically. And it's done chemically through the use of a very simple molecule called methyl group. Okay. And what these methyl groups do is they tell the cell how to use the genetic material. Okay. I'll give you a quick example. Okay. We have information in our body for making eye color. Okay. And so some people have brown eyes, some people have blue eyes. Somebody who has blue eyes, for instance, only his eyes are blue. That means that gene, that piece of information, that color, is not being used in all the different places of the body, only in the eye. And that is done by methylation. Okay? The methyl groups say all over the body the information for eye color is methylated. It has methyl groups. And only in the eye, it's unmethylated. It doesn't have a methyl group. And this gives the body information on where the different pieces of Lego, the different components of the body, should be expressed. Okay. This is a very important piece of information because this information is what allows the body in the first place to develop into an organism, to make different tissues. This information is also important in diseases. For instance, one of the major things that happens in cancer is not so much that the information is different in the cells, but rather the annotations are different. And the cancer cells are doing completely the wrong thing with the information they have. And so this additional layer of information, this annotation, called DNA methylation is a very important part of development of diseases. It provides a mechanism for us to adapt to our environment. It probably plays a role in aging. Okay? It's a very, very important aspect of the language of genetics, or as we call it, epigenetics. Okay? And it turns out that with lots of ingenuity and originality and creativity, that you can do many things once you have this information. And that's where my good friend and colleague, Yuval, comes into the picture.
Yeah, thanks. Hi. So my name is Yuval Dor. I'm also a professor of biology at the same medical school as Chaim, one floor above Chaim. I joined, I guess, 30 years later. I'm interested in a topic called tissue dynamics, understanding where and when cells in the body are born and when they die. And it turned out that the fundamental discovery of Chaim that he just described, this method of annotation, of unique annotation of each cell type in the body, has a very surprising application that relates to our ability to detect diseases. And the fundamental phenomenon that we're studying here is the fact that when cells die in the body, they release small fragments of their DNA to the circulation, and those fragments travel in the blood for a short time. It turns out that if you are able to identify those specific annotations, those methylation marks on those fragments of circulating DNA, you can essentially trace the tissue origins of those fragments. So if you can identify on a piece of DNA that circulates in your blood, the annotation, the methylation mark that says this must be coming from a breast cell, this actually tells you that there was a breast cell that has died recently in the body of that individual. It turns out that this can be done in a very specific and very sensitive manner and is reflective often of pathologies. For example, elevated levels of breast-derived, so-called cell-free DNA in the circulation is in very good correlation with development of breast cancer. And by the same principle, higher levels of pancreas DNA are indicative, circulating in blood, are indicative of early stages of pancreatic cancer. And many pathologies are reflected in elevated levels of cell-free DNA in the circulation that can be identified using that principle of cell type specific marks or epigenetic marks. And this, we think, is going to be the basis for how we're going to diagnose many diseases, in particular cancer, in the coming years. And this is a very exciting direction, a very exciting field in diagnostic medicine. Right now, we're focused on basic principles of that technology, but there are also other entities, including startup companies, that are starting to really apply that to real life. And this is really an exciting journey that I think takes us from really fundamental discoveries made by Chaim over the last several decades on how the organizing principle of life, and with this surprising application that can be used for an important task such as early detection of disease. And this goes even beyond cancer to early detection of Alzheimer's disease and additional conditions. So really very exciting.
Yuval, are there any indications that this type of test, blood test, actually can detect cancers early on, even before they're diagnosed?
Even before they're diagnosed right now. So yes, it turns out that only for a few cancers we have right now biomarkers for early detection, like mammography in breast cancer, or PSA for prostate cancer, or colonoscopies. In many cancers, there are no biomarkers, and the earliest indication of disease is typically at a late stage. For example, in pancreatic cancer, and there's already pretty strong evidence that at an early, that is operable stages of of cancers, in many cases, there is already information released to the blood that can be detected by identifying those accurate indications of cell identity or the cell of origin. Very exciting demonstrations that this can actually work. So the hope is to kind of de-stage, that's the professional term, to identify cancers at an earlier stage that is still when the tumor is still localized and can be removed.
So this is going to save a lot of lives, it seems to me.
This is the hope of many, many people right now. And really it came as a surprise to many that the method of choice for identifying the tissue origins of cell-free DNA and therefore diagnosing cancer turns out to be detecting those methylation patterns of those annotations that you have discovered. So this is really exciting and satisfying.
Wow. Wonderful, wonderful work. And as you say, very exciting.
It is exciting.
I'd like to add something here that I think is very important. It has to do with the ICRF. When I started my work a very long time ago here in Israel, I had decided that I want to work on basic biology. For those of you who don't know what that means, it means that I was just plain curious to know how biology works. And there was nothing on my mind about curing diseases or making an agricultural breakthrough or getting rid of viruses or anything like that. Nothing, making people more healthy. It was not my intention. My intention was to learn pure, basic science about how biology works. And along came the ICRF, which was devoted to treating and solving the problem of cancer. And they saw my work and they said, and this is not a simple thing, they said, we understand that if Chaim Cedar will uncover the secrets, the basic secrets of how cells work, that can be applied to understanding cancer. And they supported me for many, many, many years. I must say, any normal fund would be disappointed because I didn't cure any cancer. I didn't treat any cancers. I didn't early detect any other diseases. And it turned out in the end that the ICRF had the right approach. They understood biology much better than many, many other funding organizations. And they realized that the way to handle the problem of cancer is to first understand it. And I give them a lot of credit. And I just want to add one more thing to that, which I think is important. And that is that we're very commonly we think about treatment. A lot of money goes into treating tumors that already are present, to stopping them from spreading, or to trying to kill them, or to trying to slow them down. Right? Where the goal of medicine is not really that. The goal of medicine is to try to prevent these diseases. And this is the place where basic research comes to the fore. And again, I give all the credit to the ICRF.
I joined Chaim, and I must say that I'm also a happy recipient of an ICRF Career Development Award, Pancreatic Cancer, the Barbara Goodman Career Development Award in Pancreatic Cancer back in 2005. It was very important support. I think one of the first grants that I ever got. So I'm also grateful, but I think that the approach of supporting basic research as the most important thing towards translational research is important and unique and proves itself.
Thanks for your attention.
Thank you.